All right, 17.3 and 17 through 17.5. So here we're going to talk about gene control. Now, with gene control, we have an RNA polymerase, and I already talked about how it binds to the TATA box and makes a copy of DNA. The mRNA has to do more processing. If you saw in the last one, I put mature mRNA. We could always call the first, I don't want to say first try, but the original copy, it's immature. I don't know. All right, so here we go. We have these SNRPs, they're called. They are small nuclear RNPs. And what we're going to do is they are going to splice out. So we have them all together. It's called a spliceosome. And they are going to cut out pieces of the RNA. Notice here we have our exon here, and an intron. We need to cut the introns out. I always think of them as interrupting introns because they're interrupting exon 1, exon 2, which is the actual information we need to make the protein. This allows for what we call alternate splicing, okay, using these spliceosomes. We think those SNRPs actually are part or come from parts of introns, where we used to think introns were just junk. We're now finding that, ooh, no, they actually have, some of them have a purpose. We also have RNA with enzymatic abilities called ribozymes. So when we talked about proteins, I said like 98% or are all proteins. Well, here's that extra, that few little percentage, and that acts as an enzyme, but instead of being made up of protein, it's made up of RNA. Ribozymes are found in both eukaryotes and prokaryotes, suggesting that RNA came first in evolutionary terms. So that was the big debate, which came first, RNA or DNA. And since RNA has that enzymatic capability, it is really kind of led the way to say RNA was first. So here is our immature. So here's our immature, well, this is our DNA actually right here. And then this is our primary or immature messenger RNA. Now, with those spliceosomes, we can have what's called alternative splicing. So we could put exon 1, 2, 4, 5, 6, or 1, 3, 5, 6, or 1, 3, 4, 5, 6, which was kind of news because once we had the Human Genome Project and they found, only around 10,000, I think it was around 10,000 genes, and yet they know we have by far many more than that. This is what was discovered, is that those interrupting introns can be cut out and exons couldn't be spliced together differently to give us different proteins. So the same gene can give us different proteins based on this alternate splicing. Now, after we get our interrupting introns out, we're still not done. All right, so we can see here we have our introns all cut out, but we also have what's called a 5' cap. All right, now our 5' cap, it is, and you'll see there's a guanine up here, which is why sometimes it's called the G cap. But the 5', because it's on the 5 end of that messenger RNA cap, has different sequences based on whether that messenger RNA will go to it'll all go to a ribosome, and then it instructs the ribosome either to attach to the rough ER, which means that that protein is most likely going to stay inside the cell, or it needs that nonpolar environment to fold properly, or to go to for the RNA, the ribosome to stay free in the cytosol. All right, so that's kind of our directions. Go to a ribosome. That ribosome is free or attached. The poly A tail is kind of like a time, like a ticker. Okay, there are enzymes in our cell that will start breaking down that messenger RNA here. So the longer the poly A tail, the longer that messenger RNA stays functional. The shorter the tail, the quicker we start to get into eating the actual instructions for that protein, and it no longer works. And there's certain proteins we do not want to keep around for a long time. For example, adrenaline. Adrenaline is useful when you're in a fight or flight situation, but you don't want that particular protein to stay there for any length of time, because it will put stress on your cardiovascular system and could give you a heart attack. All right, so it's not good to keep around for a long time. Now, if you need more adrenaline because the situation's still there, then they'll make more copies of messenger RNA. So that's a great way that we control gene expression. So here you can see the G cap, the poly A tail. We've gotten rid of the introns, and now we're good to go. Okay, so basically here I just said what I did in the last slide. Translation. We're going to read the messenger RNA and put our amino acids together. We're always going to start with the start codon, which is AUG, which codes for methionine. The small ribosomal subunit will bind to the messenger RNA. And then once the tRNA for the start codon MET binds, so you can see our MET here, it's going to bind to, there's our anticodon. Here's our codon. Notice how they complementary base pair here. So they know that's the proper fit. We've got the right amino acid that the tRNA is carrying. This is going to set the reading frame, meaning this is where we're going to start reading our messenger RNA. And then the large ribosomal subunit will snap on. Now we will have 3 sites. We have the E site, the P site, and the A site. So there's 3 binding pockets inside that ribosome. Now tRNA is a single-stranded RNA. Notice the hydrogen bonds here to give it its dimensional shape. It does look like a cross, right, or a T, but it also transfers the amino acids to, you see the amino acid up here, to the ribosome. So the bottom loop right here is the anticodon, which is going to base pair with our messenger RNA. So each tRNA carries an amino acid at the top, and it brings it to the ribosome. Now here we have a special protein, this aminoacyl tRNA synthetase, and it is going to use ATP to basically reload our tRNA. It's going to bind in that pocket. It has and there's many of these for each type of amino acid, and then it's going to put that amino acid on the empty tRNA once that tRNA has delivered the amino acid to the ribosome. Now we're going to have what's a process called elongation. All right, so we had our beginning, our start. Now we've got an oh, I can't think of the word. Let's just say make our messenger RNA longer. And so we have that first codon here in the P site. This is P for peptide bond. And then we have our A site for acceptor. Okay. So as we read the next base pair and the tRNA pops in, you'll get them popping in, popping out, popping in until they get the right fit. Once they have the right fit, you are going to make a peptide bond, all right, between the 2 amino acids, and then they're each going to shift over. So this is I always call it the empty, the E empty, the tRNA is now empty. It's going to be released. Now we open up another acceptor site and the next tRNA will pop in. And we're going to continue this process until we get to termination. So you see, as we put all those the polypeptide here with all of the amino acids together. So once we move over, we now have a sequence for a stop. The stop does not fit a tRNA in there. It's a release factor. Now, once that release factor binds, what happens is it goes to pass off the polypeptide chain, but there's nothing to connect that peptide bond to. So it releases, you see how it's released, the protein here, and the large and small ribosomal subunits break apart. We have this thing called the third codon. It really should be the third base. Ooh, let me change that. The third base in the codon wobble. Okay. So there are approximately 40 different tRNAs. This is because the third base in a codon does not always follow the base pairing rule. Notice in the chart, if you change the third base, it doesn't always change the amino acid. But what happens when you change the first or second? Okay, so notice here, I could change the third base in that codon, and I still, let's not do that one. That one does change. Right here. Notice they're all serine. Same with here. All proline. All right, here, all alanine. All right. So that's that third base in the codon, doesn't generally change the amino acid. Now, if we were to change the second base, okay, so if I was to change, let's see if I can find one here. All right. If I was to change the second, let me get rid of it. Okay. Nothing's popping out at me because those are the same, and then we have two different. But let's just say here, let's just say I changed this to a G. Okay, alanine is no longer going to be alanine. It's going to be GGC, and we're going to get glycine. So it happens with the first one too. If you change the first or second base, you're going to change the amino acid. Now, we have what's called polyribosomes. So when you are making a copy of messenger RNA, you can have many ribosomes reading that messenger RNA at the same time. So they're just kind of following each other down the instruction set, the messenger RNA. Now, polyribosomes in prokaryotes are really kind of cool because prokaryotes can have transcription and translation occurring simultaneously. So we have our DNA here. We are making our messenger RNA. And then we've got our ribosomes, many of our ribosomes, polyribosomes. And you can see kind of the cartoon here as we're putting out the mRNA, the sort of transcription. We're immediately translating. And so I'm going to ask you, oh my God, keep going. How can this happen in prokaryotes? Why can they do it? We can't do that. And the reason is because of the nucleus. DNA replication and transcription occurs in the nucleus. Remember, eukaryotes have that compartmentalization. So as soon as the mRNA does leave the nucleus through the nuclear pores, we are able to translate and make those proteins. Now, the last section is about mutations. Okay, so here, this is the normal. All right, so our DNA is TTC, our complementary messenger RNA is AAG, and that is going to give you the amino acid lysine. Now, remember how I said that third base could change, and we still generally get the same amino acid, which is why it's called a silent mutation. Then we have what's kind of nonsense, and again, that's generally like the first base here, and it changes the amino acid to a stop. So you will prematurely stop making your protein, which generally that protein's not going to function. Now we also have a missense, and missense, you could have a conservative or non-conservative. So a conservative means we did change, we had a change here. Okay, notice we have a C change that gives us a G, and we're going to get arginine. Now, arginine, these are color coded based on what type of amino acids they are. So, are they positive? Are they negative? Are they polar? Are they non-polar? And notice this one is positive, and so is this one. That is a conservative change. That may not change the way that protein folds there because it has the same characteristics. Therefore, it may still function fine. Now, notice the color of this one. The side chain is different, and that is threonine. And it has different characteristics. And generally that can, I'm not gonna say it's always, but that can change the enzyme, or not enzyme, but the protein, it's folding, and therefore it will not be functional. Of course, we know, like, for example, enzymes have an active site. So if this change occurs somewhere else in the protein and we can still have our active binding site intact, it may not change the function of that, that protein either. So it really kind of depends on not only if it is a totally different characteristic, it may also vary on where it occurs in the protein. Okay. We also have what's called a frame shift mutation. So if we were to insert or delete a nucleotide, it will change the reading frame. Remember, our methionine is going to set that reading frame. And so with this example, okay, with this sorry, my wrist was sore. With this example, remember we read nucleotides 3 bases at a time. That's our codon. So if we make this sentence, the fat cat sat, if we were to delete that first nucleotide, everything shifts. And if you see that, what is it? I don't know, it makes absolutely no sense. Everything, what we call downstream of that insertion or deletion, will totally be different. And it will definitely not allow that protein to function correctly. So here's another example. So we have our deletion here. And the same thing will happen if we insert, right? Whether we insert or delete, it's going to affect the reading frame. So we have here serine, threonine, glutamine, phenylalanine, and asparagine. When we shift that, instead of reading it ACG, we're now going to read it CGG. And notice we start with our correct amino acid, but everything downstream of that, either insertion or deletion, is different. That will definitely mess up the protein. Okay. So hopefully, you can understand a little bit more about transcription, translation, and mutations at the, I would say, molecular level. We're talking about DNA errors that could have been made. All right. I hope this was helpful, and I'll see you in the next video.